So Athena is our software platform for analyzing the images out of the Hermes, and it is enabled for real-time analysis, meaning that as the images are being acquired on the Hermes, you can set up Athena to begin analyzing them at the same time. So the way that works is, again, through a touch button interface. So these are just screen clippings, but the layout is very similar to the Hermes software. And as you change any of the parameters, so in this case, we're changing the threshold to identify the cellular cytoplasm, we can see an update for the segmentation masks that are identifying the outline of the cells. If you've done this using ImageJ, then many of the same types of parameters will appear and give you this visual readout to see what the computer is able to detect given the input parameters. If you have trouble or challenges identifying the right parameters, where we would be happy to help to get you started, because once you have the parameters defined, you can save them as what we call a protocol file, and that protocol file will automatically define the parameters so that you can reproducibly analyze your images using the same settings. As I mentioned previously, once the image analysis is finalized, then you're going to get a lot of data visualizations. So you get heat maps. This basically will take one of the attributes at a time across the multi-well plate and give you a visual and colorimetric readout for the range of values that are extracted from your images. If you uploaded a tags file, then the software knows the concentrations and treatment conditions across each well and can estimate a dose curve. If you do a time lapse, then you can look at the time evolution plot of any of the parameters that you want to extract from the images. As well, we have a 2D scatter plot where you can plot two attributes versus one another and try to look for outliers or different types of behavior within your sample type. What's nice is that all of these images and the underlying raw data that's displayed within them can be saved at the touch of a button. All of these images can then be put directly into PowerPoint files if you want to make a presentation or you have a digital notebook. And the underlying data is saved as a CSV or comma separated value file, which is easy to upload into Excel, MATLAB, Origin, GraphPad, or any other software that you may be interested in using for downstream image data processing.